my presentation here. I will just start my to share my screen. Okay. Can you see my screen with the presentation?
Perfect. Please go ahead.
Thank you. So my name is Liang Qiao from Fudan University, Shanghai, China, and it's my great pleasure to participate in the Future Developers meeting. My presentation today is about the DIA proteomics with in silico spectral library by deep learning. Because I have only 10 minutes, so I skip the glycoproteomics DIA, which will take much longer time. So as we all know during the week already about the DIA, it features with the collection of the MS2 independent from the MS1. So in this way, we can collect almost all the fragments of all the precursors that are inside the sample. And due to such a feature, we can get exactly the MS2 extracted current chromatography for quantification, which is normally more accurate and provides a dynamic range wider than the MS1 based quantification. But the problem is that for the DIA data, we have a very complex MS2 spectrum, which includes the fragments from many different precursors, making the analysis of the DIA data more difficult compared to the DDA. So the typical strategies include the peptide-centric DIA data analysis and the spectrum-centric DIA data analysis. In the peptide-centric DIA, we firstly pull all the samples and then perform the fractionation. And then for each fraction, we can perform the DDA analysis to generate at the end of the experiment a deep coverage identification results to build the spectral library, which includes the retention time of the peptide and also the MS2 profile. On the other side, we collect the DIA data of each sample, and then we query the DIA data against the spectral library for identification. The second strategy about the spectrum-centric DIA, normally we firstly group the precursor and the fragment depending on the elution profile similarity. And then we can generate this kind of pseudo MS2 spectrum for each precursor. And then we can do the database sequencing searching, which is rather similar to the DDA analysis workflow. In our strategy, we use the peptide-centric, but we use the deep learning to predict the spectral library instead of generating the spectral library by DDA experiments. So this is the deep neural network we use for the spectral library building. We have the input part, which can convert the peptide sequence into a two-dimensional matrix. So for each column, we have a amino acid, and for each row, we have a position of the peptide, of the amino acid in the peptide. For instance, here, the first amino acid is an F, so we fill this cell with 1 and we leave the others as 0 to convert the sequence into the matrix. And then we have the convolutional neural network and the recurrent neural network. And at the end, we have the output. We can output the retention time, the best phase voltage for the peptide, for the precursor, for the peptide or the precursor, and also the Tim's and mobility for the precursor, the peptide detectability, and also the MS2 fragment intensity. So normally we use some experimental data to train the models first, and then we can use the pre-trained model to perform the prediction. Then for any given peptide sequence, we generate directly all these values here. To train the model, just to give you some idea, we use normally kind of 2D LC mass DDA data of a cell line, which is enough to train the model. For instance, here it includes the PSMs from 46,000 peptides, including 38 doubly charged precursors and 80,000 triply charged precursors. For each precursor, we keep only the best PSM. And for the model training, normally we use 24 hours on a normal workstation. Then we evaluate the performance of the prediction on different data sets. So this is about the MS2 prediction. We compare the dot product between the predicted MS2 and the experimental one. So these are the results by the deep DIA. These are the experimental repeats, and these are the results by other prediction methods, including PDEP, ProSite, and MS2 PAP. So basically, for deep DIA, it can predict MS2 spectra very similar to the real experimental ones, close to the performance of the experimental repeats. And also, we can see that the lab-specific models outperform the others. So what is the lab-specific models? That means we use the data generated from the same lab to train a model, and then we use the model to and then we test the model on the other data set also generated by the same lab. So for example, here we use the HALA one to train the model, and then we predict the mouse sequence and compare with the experimental results from the same lab. And it works better than the remote ones or the closed lab ones. And similarly, we can also do the IRT prediction. And basically, the correlation of the predicted IRT by deep DIA and the real IRT is larger than 0.99, so it's here. And it's similar to the experimental repeats compared to the other prediction tools like ProSite and SSR Calc. The performance is also better here with the deep DIA. And for the others, we have also performed the detectability prediction. So what is the detectability? Actually, it is to generate a list of peptides that are likely to be detected by the bottom-up proteomics methods. And basically, we have the detectability score, which is based on the relative quantity of the peptides from the MS1 feature. If the peptide is only detected with fractionation, we define the score as 0.5. And if it can be detected without fractionation, the score is from this equation here, and normally it's between 0.5 and 1. So we also train the model and then test it. So if we put a threshold of 0.5, we can have a recall of 0.77. That means 77% of the peptides detected by DDA experiment is also predicted by the model here. But the predicted list also includes almost 20% of peptide that is not detected by DDA. But for DI analysis, because we can control the FDR during the analysis, so it is more important we have a large recall, even we have some we can see error inside the predicted list. And similarly, we also predicted the air mobility by timsTOF, and the correlation is not bad. We can see here for the doubly charged precursors and the triplet charged ones. And for the FAMs, we can predict the best voltage for a precursor, and the red ones show that the prediction agrees with the experimental results, and the blue ones, that means the prediction disagree from the experimental results. So with the prediction, we can then generate the spectral library, and then we use the predicted spectral library to analyze the DIA data. So here we use a HALA data set, which is from the QEHF. So we use 3 different protein sequence database. The 6K means proteins identified from HALA by DDA experiments, and 10K means the pan-human, and 20K is the Swiss prot. So basically, we perform the in silico digestion, and we keep peptides with the miscleavage equal or smaller than 2, and the detectability threshold is 0.5. The dotted line means the ID by the DDA library. The green one means the shared identification between the deep DIA and the DDA library-based. The blue means that the peptides or proteins only identified by the deep DIA, and the right is identification from the entrapment sequence, which is basically a protein sequence from microbeads. So we can see that with the pan-human, we have more identification in the protein level and the peptide level, and the entrapment percentage is a bit more than 1%. And we also test on mouse, and the performance similar to the HeLa. And also we test on human serum. So here we use 3 human serum samples without high abundance protein depletion. So for the DDA library, the sample-specific DDA library, we have 7.5 thousand peptides from almost 1,000 proteins. And for the predictive library, we have a lot more peptides and proteins, which are from the previous projects in our lab, because we have a larger library here. So for the deep DIA, we have larger number of identification, but we need to be careful to make sure that the identification are correct. So we use the isotope labeled peptides to estimate the FDR. So basically, for different methods, the estimated FDR is lower than 1% by using the isotope labeled peptides, and these peptides covers from the high abundance to the low abundance. So that suggests also the estimation is close to the real case. And recently, we also make some tests on the FAMS Explorers instrument and the TEAMS instrument. So basically, for the FAMS, our performance is close to the DDA based. And also for Spectra notes, recently it released the direct DIA 2.0. The performance is also very good. And if we combine the two together without building a library, we get already quite good number of identification. For teams, we also make some tests, but at this moment, the performance by prediction is not as good as the DDA based. I think because maybe we don't have very good prediction on the mobility and also MS2 for TOF. So at the end of the slides, there's some key message. So we suggest to use some lab-specific models for the prediction. That means you can generate the data by yourself, and then you train your model, and then you can use the model on the same instrument. Because from different labs, we may have different sightings to the instrument, and that can bring some difference to the MS2 and the retention time and so on. And also we should take advantage of the public available peptide list, but using the DNN models to regenerate ART and MS2 and also the other features for data analysis. And also we think the method can also be useful for PRM and DDA. I would acknowledge my students and colleagues, and also thank you very much for your attention. And I think we leave the question and answer at the end of the section. Thank you.
Thank you so much for the very interesting presentation. Let us all give Dr. Schauer a virtual round of applause. And their work generates quite a few questions. Unfortunately, we're out of time. So Dr. Schauer, if you don't mind answering the questions in writing, just by typing in the Q&A.
I will write in the Q&A.
Yes, I think this will be the best. This way we can move on, but please do not hesitate to ask more questions. The kind of one of the few advantages of the online format is that we can continue answering questions in writing, and you can continue asking them. So please don't be shy. And with that, let us move on to the next presentation. So our next speaker is Behruz Shamisei from University of Cincinnati. And Behruz will share with us his work on PyNet. I'm looking forward to this presentation. Welcome, Behruz.
Thank you. So I hope that you are seeing what I'm looking at, which is...
We do.
Okay. Thank you, Dr. Witek, for putting this great two works of presentations for all of us, and thank you for giving me the opportunity to present PyNet. PyNet is coming from the Links project, which is analyzing the effect of perturbations on the proteomics and RNA-seq readouts. So the input for PINET is already processed peptides and proteins. And PINET is designed to first to analyze the links proteomics data, but it is a versatile tool that can be used for other data sets, as I will show. So I had this 10 minutes, so I decided that it's not that much time. Let's have why cannot, okay, cool. Let's have a demo of PINET. So this slide shows the features, what PINET does, and I will go to give a demo of how we can use it. So we published PINET last year, basically last year, May of 2020, in Nucleic Acid Research, a big consortium of links proteomics centers. So University of Washington were involved, Mike Mackos, Brandon, you heard from him last week. So we presented PINET, I think it was like 2.5 years ago, in University of Washington, and we got a lot of feedback from people from Broad Institute, especially Jake Jaffe, and it came to this product. So there are a couple of key features that PINET does. That's PTM centric analysis, connection between PTMs to enzymes. We analyze peptides, we map it to proteoform, and look at the proteoform data. So I have embedded fast peptide search, how it can be quickly mapped to proteins, which I will show. Versatile data visualization, and connection to different resources, in the gene level data. It would be better to show you, how it works. So PINET, if we go, that's the URL, pinet-server.org gets us to this landing page. It has 3 workflows, as you can see on the top. The first one is peptide to protein mapping. So the data should be already processed. We should have the peptide level. Just peptide would be enough. But if we have expression values, that would be better. So after mapping peptides to proteoform, we will have 2 workflows afterwards. One of them is from PTM to modifiers and protein to pathway. So depending, what is the proteoform from the peptide. I have embedded multiple organisms, so we can, the input peptide based on the library of selection can be mapped to, the different libraries, and also UniProt, SwissProt, libraries. So one of the assays that we had in links is the P100, which is 96 phosphopeptides, which I click here and submit, and you see, it took a couple of seconds, or less than a second to map 96 peptides to proteins, and this mapping can be visualized, and we'll see the connection. The peptide phosphorylated and theorine at site 610 is found, in this protein. So we can select and see how is the connection. Again, the peptide it is found in the protein Q15149, and we have 3 layouts for this, because the one peptide can be found in multiple proteins, and based on the selection of these 3 tabs, it can be, the readouts can be assigned to one representative protein, which is razor protein, or those values can be averaged to multiple proteins, and you can mouse over and see how it is getting averaged. The information about the PTMs also, we have embedded those. So in different tabs that we can select, we can just click and see what are those PTMs. Again, multiple tables to see the connection between peptides and proteins. The same thing that you visually saw there. Also, where these peptides are found, we provide different graphs to show, the cellular location, gene ontology about those peptides. So, here we had 96. We may have, much more if in the file submission. So we have embedded the MaxQuant file submission. So, for example, we can use. I have embedded the evidence file from MaxQuant, so it can be downloaded. Again, it can be submitted to show how this works. The organism for this example is zebrafish, so we should be checking the zebrafish for the library. The control and the treatment, it should be assigned here in these two tables, because in the MaxQuant, basically we will do t-test between two conditions, and based on the log fold change and the p-value, we select the peptides for further analysis. Some peptides do not get much of expression change. There are also different normalization, different data imputation that we can select here. As you can see, it is now it is getting ready for roughly 1,400 peptides. And that's, I would suggest that for this tab just be around less than 2,000, because it gets overwhelmed. The server gets overwhelmed here. That's the result of the t-test. We can see that these peptides can be selected based on the p-value. And that changes, this selection changes, the selection underneath. And the graph underneath here, which is dynamically changing, will change the input for the second task. For example, if we select these peptides, the PTM and the protein level expression values will change in the second and the third tab. Now I went to the second tab. The organism automatically assigned here, but I will select P100 peptides for this one, because the input file here, it can be coming from the first tab, or it can be assigned in the second tab, different PTM. As I said, in the second tab, the connection between enzyme and the PTMs are shown here. Every edge, every color is explained in the question mark. For example, here we had low expression of one peptide, and we can see what are the enzymes that are connected specifically to that protein phosphorylating that exact site. It can be viewed differently. We can, everything is dynamic here in these figures that can be adjusted with those bars on the top. So what it shows here is a kinase that specifically phosphorylates one peptide, one protein at a specific site. And this is not only for phosphorylation analysis, we have different modifications here. Another tab, this is about deep phos, which is deep learning phosphorylation, kinase phosphosite correlation, that you can select this score to make sure that we have a high probability of one kinase is phosphorylating one peptide. Again, I'm going very quickly here. Mouse over the peptides shows the connection. I go to the third tab. This is where the protein level, the gene level data, we do analyze the gene level data. Again, I go to illustrative example, or we can go result from the peptide to protein tab. For the illustrative example, again, this is P100 assay gene level data. We will see the information about the genes. The pathway enrichment, that's the gene level data. This is enrichment analysis, via enricher, but we added with some added functionalities here. For example, we do highlight the pathways. We can select those pathways, and enricher group is also part of PINET, are also part of PINET. Again, the different types of visualization. Every graph that you can see here can be selected dynamically and can be downloaded with high quality images. And the last thing that PINET does is connection to links, which is the essence of PINET. That basically, if we generate a signature from the first tab, from the second or third tab, how can we find some drugs that reverse this signature, or what are the gene knockdowns that their readout is similar to our input. Again, we can see that these are the drugs that in some cell lines, that's the red means concordant, the blue means discordant to our input signature. I'm going very quickly over all of this. We have explained everything in detail in the help and in the actual paper. I want to show that it can be downloaded as high resolution images that it can be used in the research. In both can be downloaded in PNG and SVG, all of these graphs. Again, a lot of information can be found in the help, which I will be going to show you about the PINET, about the workflows that I just showed you, what is done in each workflow. Everything can be seen and, it is explained over there. All of these API calls that that PINET is using, and we are embedding, I've embedded that's here, the examples are here. For example, this is just to search, where these peptides are found. It can be used via API. We can generate the same graphs, with user input CSV files and acknowledgements about all of the tools and resources that have embedded in PINET. That was very quick, for the follow-up questions. I put my email here. I want to again thanks Dr. Vitek and all of the the groups that we had been working with in the Links consortium to get this done, and I be ready for the user community.
Thank you so very much, Behruz. Let's give Behruz a virtual round of applause. Thank you for the presentation. We have several questions to you, and also in the interest of time, if you don't mind answering the questions by typing in the Q&A. And to the attendees, please continue sending your questions. But if you don't mind, add the name of the speaker to whom the question is addressed so that it's easier to navigate as the questions kind of start accumulating.
Thank you.
But please don't be shy. So this is the advantage of the online forum, that we can actually be quite extensive with the questions, and we very much welcome that. And so our next speaker is Ahmed Youssef from Boston University. And Ahmed will speak about investigating interactome dynamics using CFMS data. Welcome, Ahmed.
Thank you, Professor Vitek. Hi, everyone. My name is Ahmed Youssef. I'm a PhD student at Boston University, where my research focuses on protein interaction networks. But first, I just want to say thank you to Professor Vitek and everyone for organizing this great meeting. I've learned a lot these past 2 weeks, and thank you so much for organizing it, especially in this virtual format. And of course, for giving me the opportunity to talk about my research today. So, as this title slide suggests, my research is interested in protein interactome dynamics, and today I'll be talking a bit about some of my ongoing work in developing computational methods for studying interactome dynamics from co-fractionation MS data. So first, I just wanted to give a brief introduction. So out of all the areas of proteomics, my research is interested specifically in protein interactions. And to be even more specific, looking at the physical contacts of proteins. So for like this example protein complex I'm showing on the top right here, where we have a group of proteins physically in contact with each other. And to bring this back to computational side of things, we usually present these interactions as a network like the one on the bottom right here, where each node is a protein and each edge is a physical interaction. Now, if we were to take all such interactions in an organism and put them in one large network, we get what is termed the interactome. And on this slide, I'm showing an example from one of the early influential papers in this area where they profiled the whole interactome for the fruit fly. Now, the main motivation for studying this interactome in our lab, as opposed to individual interactions, is that there's evidence in the literature that most human proteins function in the cell, not in isolation, but rather as members of these protein complexes. And this is actually related to the talk by Isabel last week, where she was also talking about something similar, looking at protein complex using co-fractionation data. Now, the particular aspect of these protein interactomes that I'm interested in studying is this idea of network remodeling. And the premise here is that these interactomes are dynamic systems that can change across conditions. For example, like in this toy data I'm giving here, you can imagine that in response to a disease or some kind of change in the organism's environment, the interactions that occur between the proteins could change in some way. They could be disrupted, you could have new interactions forming, and so on. And so if we want to understand, if we want to reach a full understanding of how the protein interactome actually functions, we need to study it in terms of its dynamics rather than in a static manner. And on this slide, I'm just giving a few examples of diseases that are linked to this interactome remodeling. And now each one of these are interesting case studies, but in the interest of time, I'm not going to go over them. But this is just a way to motivate why we actually care about this phenomenon, is because it is directly related to multiple diseases. So now I want to talk quickly about the particular experimental strategy that we have in our lab for identifying these protein interactions to begin with as the very first step. And we use co-fractionation MS, and on this slide I'm showing a figure from one of the early papers in this area that also came out from our lab. And very briefly, what we do in this experiment is we start with our native protein sample, and we use HPLC to fractionate the proteins under non-denaturing conditions to keep the complexes intact across multiple fractions. They're separated based on size or charge. And afterwards, for each one of these fractions, we use standard LCMS to quantify the proteins within each of these fractions. And then from here, we take a guilt by association approach, where the idea is that the proteins that elute together in the same fractions are more likely to be members of the same protein complex. Okay, so this is the experimental workflow, but what does the data look like for me on the computational side of things? Well, here's an example, a protein profile that comes from this experiment, and this is from a data set that was generated in our lab that I'll talk a bit more about later. And in this example, you can see on the x-axis we have those multiple fractions I mentioned earlier. And again, this is an earlier fractionation step, even before LCMS. And this is the intact protein samples before trypsin digestion and all of the standard sample prep. Meanwhile, on the y-axis on this plot, we have the intensity as a measure of abundance, basically. And as you can see, for this protein, it shows up in a small number of fractions, forming this kind of peak shape, which is kind of typical for this type of data. Now, if I were to take several such proteins and put them on one plot, we will get something that looks like this, where you can see good overlap among these peaks, which suggests that we have a protein complex or some kind of interaction occurring among these three proteins. And an interesting thing, you see there's a second blue peak here for this protein Hall A, which suggests that it might be a member of a second protein complex, which is one of the advantages of using this experimental workflow. Okay, so now to bring this back to our research question, we're interested in studying remodeling. And the way this would translate to this type of data set is that you can imagine, to follow up with the previous example from the previous slide, if we had some kind of environmental perturbation, this could affect our proteins in a way that, as you can see on the right, the proteins do not overlap anymore. And this decrease in the overlap suggests that this interaction might have been disrupted in response to this environmental perturbation. Now, the other scenario we're interested in is that the interaction might still be intact, but it might have decreased in intensity. So if we see on the right here that the change occurring is a change in relative intensity rather than some kind of shift where the proteins move away from each other, then this could suggest that there is less of this protein complex under these conditions, which could tell us something about the relative importance of this complex within this experimental context. So these are the two patterns that we are looking for in this data when we're talking about interactive remodeling. And this brings me to this pipeline that I'm currently working on to find such cases of interaction. And hopefully, this is still a work in progress, but hopefully we can get this submitted for publication soon. So this is a large pipeline. I'm not going to go into too much detail on all of the steps in the interest of time, but you can see that's split into two main modules. The first on the left is the boring data preprocessing. So we have multiple steps here attempting to have a tailored strategy specific for cofractionation data, which is inspired by some of the other tools that are out there and some of the other work looking at how can we work with this cofractionation data, because it has its own challenges compared to kind of your standard shotgun proteomics experiment. But then once we've gone through that and created our protein profiles, like the ones I was showing in the previous slide, we can then move on to scoring the remodeling that we're interested in. And here I'm showing the three main steps involved here. And the first, I'm sorry, I was looking at the chat. So in the first box here, the first thing we are doing is we are scoring those two incidents of rewiring, and those two different types of scenarios I was mentioning earlier. And we have multiple distance metrics for this particular step. The next step here is to look at how these profile similarity distances are changing across conditions. And this is our measure of remodeling. In other words, do these protein profiles seem to be moving away from each other or to go towards each other, or is there some kind of coordinated change in intensity? And now, given such scores, which in this kind of example I'm showing using a heat map, we can then take a reference PPI of interest or any set of complexes we are interested in, for example, a set of complexes from the quorum databases or any existing reference interactome, and we can search through this interactome to find evidence of remodeling within each different pair of conditions. So to zoom in a bit on this last step, I have kind of blown it up in this slide. And so the key idea here is that we are working with a given reference interactome. So the strategy that I mentioned is scoring each given PPI within each given pair of conditions, and it scales to multiple conditions. It does not have to be two conditions. And we are ultimately limited really to whatever reference network we are given. Now, if we want to find de novo interactions, we can just simply score all pairs of proteins if we have the computational resources for that. But essentially what we are doing in the standard strategy is we're taking our PPI, going over it one interaction at a time, scoring the remodeling that's occurring. For example, on the top here, you can see an example where under one condition, the protein profile showed good overlap, and in the second condition, they move away from each other, suggesting some kind of remodeling. While on the bottom, we see another situation where it's more of a coordinated shift, where the two peaks are moving together to the right. And looking for these kinds of scenarios, we can ultimately find those pairs that show some evidence of remodeling between our given conditions. Okay, so now I just wanted to introduce a particular case study to try to illustrate how we would actually use such strategy on a real data set. And in this study, we're looking at E. coli interactome remodeling in response to environmental perturbations, where we have E. coli grown under different experimental conditions. And the question we want to answer is whether we can come up with a strategy to find changes in the protein interactome across these different experimental conditions. So on this slide, I'm just showing a brief cartoon of the experiment that was carried out in our lab. And in this case, we have E. coli grown under 10 different growth media, where we varied things like the temperature or the sugar source, etc. And as I mentioned on the earlier slide describing the co-fractionation workflow, we extracted the proteins from each one of these growth media. We did an earlier step of HPLC fractionation. In this case, it was ion exchange chromatography to give us 96 fractions for each one of these 10 conditions. We then digested these proteins into peptides using trypsin. We used TMT labeling to multiplex our experiment, and then we finally performed LCMS to identify roughly 2,000 proteins in each one of these 10 growth media. And for each one of those proteins, you have this kind of a fractionation pattern over the 96 fractions. And what we did really to extract meaningful results here is we started with a reference interactome, which I compiled both from empirical evidence as well as external data sets. So these are PPIs that are reported in the literature. And we scored these PPIs based on their changes in interactome strength across conditions. And using this strategy, we were able to identify a smaller subset of this interactome that shows evidence of extensive remodeling in response to changes in the growth media. And now this is there's many results to pick out of here. I'm actually still in the process of picking through those results and trying to get the best story I can out of here. But because this is such a data-first study, I've also released alongside this tool a interactive web app, and this is using RShiny to visualize these results in interactive manner. So here I'm just showing a screenshot from the app, which is live now, where the user can take a set of proteins they're interested in and see how their overlap changes in the different environmental conditions. Or given a protein of interest, what you can see on the right here is looking at how that particular protein's interactions look like in different conditions, or each one of these green lines represent a different condition. And again, this is an app that is live, and we plan to use this tool not just to refine the results from the E. coli dataset I introduced today, but also to apply it to other co-fractionations datasets of the same nature. Now, I will stop here. Thank you so much for listening to my presentation. I will be happy to take any questions or comments.
Thank you so very much, Ahmed. This was very interesting. And let me remind all the attendees that please do not hesitate to ask questions in the Q&A box. And I hope that this will be useful to many. So in the interest of time, we will move on to the next presentation. But again, the Q&A box is open. So our last contributed talk of today is by Piotr Rajinsky from University of Warsaw in Poland. And Piotr will talk about Mastermind, accurate monoisotopic mass determination based on isotopic envelope. Welcome, Piotr. And we can see your screen, but we can't hear you.
Hey, hello, do you hear me? I have small problem.
Yes.
And you see my screen, right?
That's right.
Okay, so thank you. Hello, everyone. I would like to present our work about predicting where monoisotopic peak is. At the beginning, I would like just to remind what monoisotopic is, because maybe someone don't know. On the right, there is a spectrum, theoretical spectrum, so it's a perfect mathematical object. In real world, it's not so perfect. There is also a lot of noise, and even of this perfect plot, this monoisotopic peak is very small. It's the most possible left peak which can occur, and for not very small proteins, it's almost invisible. However, it's very important to classify proteins, and that's why we try to predict it better than the previous works does. And we begin with only theoretical spectra, because they are perfect mathematical objects, and that's why it's easier for the beginning. And we begin with the very simple idea. to just take average mass from the spectrum, variance from the spectrum, and learn linear model and predict just based on linear model what the monoisotopic peak is. And here is a plot of the error. And so far, this initial prediction is really, really bad. I mean, error equal to 0.4, for example, is definitely too big to be useful to anyone. However, there is one very important feature here, that 96.6 of the predictions is closer to the true peak, closer than half of a Dalton. And why is it so important? If we look at the spectrum, then we see that aggregated peaks are far from each other, approximately, not equal, but approximately 1 Dalton. So our basic idea for the theoretical spectra is that if we have prediction and we know that almost sure it's closer than half of a Dalton to the correct answer, correct monoisotopic peak, then maybe we can create such a grid that can be easily fit to the visible spectrum, visible, I mean, later in experimental spectra, but visible, and then check which point of on this grid is the closest to our initial prediction. And such grid have two features, have a distance between points and shift of this grid, and we have to estimate those two parameters. It's not so difficult to estimate grid step. We started to roll such circles through the spectra. And if we roll such circle that collects our peaks on it, then a circle which will have circumference, a circle which collect all peaks in smallest area, then it will have circumference equal to the distance between peaks. However, it takes a lot of computational power to do this. So we did this for, let's say, I don't remember, 2 million of proteins. And then we trained a linear model, also second linear model, that will give us formula for dependent on mass for these distances between peaks. Then we have also shift to take, to calculate, to estimate. And in fact, idea is quite similar, because if we have such circle and we know where the peaks are on the circle, I mean, we know that they are in small area, so the place on the circle where they are can be transformed to the actual shift of our grid. And then finally, if we have grid, we fitted it to the spectrum. So far theoretical, we can round the prediction to the closest point. And there is some correction caused by the fact that distances are not equal in every part of spectrum. For example, on the tails of the spectrum, they can be smaller. So this is just small correction. And we can take a look how it works. On the top line, there are two pictures in Dalton, error in Daltons and in parts per million, which shows how is distributed the initial prediction error. And it's a repeated plot from a few slides ago, and then adjusted model after the rounding, and we can see that it helps a lot, we almost always are in the exact place. And this is very important because this is the main problem with such algorithms, that usually the error is really close to 0 or really close to 1, 2, maybe -1 Dalton, etc. And so far, the best algorithm that predicts the monoisotopic peak is called MIND. So we compared with MIND, and I attached here errors in ppms. However, as I told, errors in ppm are not so important in our problem. The most important is how often the algorithm mistakenly takes -1 or +1 error, this shifted prediction. And we can see that we are far better so far. That's only on theoretical spectra so far, but on theoretical spectra, we are far, far better. Okay, and now we can go to the experimental spectra part. And there is a problem, because we have to measure variance and average mass from the spectrum very, very, very precisely. And it's quite impossible in experimental spectra. There is too much noise. There is too much noise even for measuring average mass precisely. And so variance is almost impossible. So our idea is, if we have experimental spectrum, we can try to fit a theoretical spectrum and check which theoretical spectrum fits the best. And on this slide, I presented an algorithm, but the main idea of everything what is on this slide is that we want to take some set of chemical formulas, which are in, let's say, 5-dimensional space of chemical formulas. We look the space for, at the beginning, for the chemical formulas that seems that have similar average mass and are not very distant from the, should not be very distant from the experimental spectrum. And then, based on optimal transport, Wasserstein distance, we check which of the spectrum fits our experimental spectrum best. And this is presented on these two plots. On the higher plot, there is a myoglobin experimental spectrum. And on the lower plot, there are two colors. The dark one is a theoretical spectrum of myoglobin, and the orange one is the fitted spectrum. So we see that this idea works pretty well. And if we fit the spectrum, we can take from it average mass, we can take variance and put to the previous algorithm to obtain final prediction. And the question what appears often is why we can't use this fitted spectrum to just take the monoisotopic peak from it. Why do we need this part with linear models? Maybe it's not necessary. We think that it is necessary because it's a safer solution, because we have no idea if the fitted formula is even similar to any possible protein. We control what happens in the center, because in optimal transport, the higher peak is more important. But because of that, maybe on tails of the spectrum, there are, maybe tails are smaller or something. But our linear models are trained on true proteins, and hence we know that that it should have, tails should have similar shapes as in usual spectra are. And here I have a workflow. This is part of learning process that gives us to linear models. And then when we have those two linear models, prediction goes to just fitting the spectrum that fits best. our experimental spectrum, take the variance and average mass, put it into linear models, and then we have final prediction. And we are almost done. We have to do more tests. So far, first test shows that even on experimental spectra, we are better than mind. We also want to test adding a voting procedure to have even less this -1 + 1 errors that can occur during the fitting spectrum. Yes, and I think that's all. Thank you very much.